Good morning. I would like to thank Scientific Committee for the opportunity to share my experimental research. My name is Annamaria Russo, and I come from University of Messina. The aim of this research is to study new synthesized environmental polymeric sensors based on polyvinyl alcohol modification by flavonoids addition, spectroscopic and functional properties. The use of biodegradable materials to produce bioplastics seems to be the only approach to reduce environmental pollution and the problems caused by fossil-based plastics. The most widely used materials for the production of bioplastics are natural compounds derived from biomass extraction, like cellulose, starch, and gelatin. Polymers derived from non-biodegradable alcoholic fermentation processes, like polyethylene. Degradable synthetic polymers, like polyvinyl alcohol, or biopolymers derived from bacterial processes, like polylactic acid and polyhydroxyalkanones. In this frame, a polymer that is a good candidate for our study was polyvinyl alcohol, PVA. It is a synthetic polymer, obtained by the partial or complete hydrolysis of polyvinyl acetate, biodegradable and soluble in water, with good thermal and mechanical properties. These excellent qualities make PVA a material widely used today in various sectors, such as medical, pharmaceutical, textile, and food. On the other hand, in the last decade, there has been a growing interest from the scientific community in natural compounds and polyphenol-rich extracts, commonly considered waste products of the food and flour industry. On this basis, the aim of this work was to produce PVA-based bioplastics functionalized with three different polyphenol, quercetin, curcumin, and bicalien, and analyze them in terms of structural, optical, mechanical, antioxidant properties to assess their potential application as a colorimetric sensor capable of detecting different environmental pollutions. The preparation of PVA-based bioplastics functionalized with flavonoids was processed using the casting solution method. In detail, we have prepared two different types of bioplastics starting from PVA in the presence and absence of starch and glycerol. In the first case, the PVA were solubilized in distilled water to obtain a clear PVA solution. At the same time, a stock solution containing starch was prepared and glycerol was solubilized in the starch solution. The starch/glycerol solution was mixed with the PVA solution, resulting in a mass ratio of, for of starch, 2 of PVA and 1 of glycerol. Subsequently, 3 separate solutions of quercetin, curcumin, and bicalien at various percent were added for each bioplastic. Finally, 10.0 milliliters of each prepared solution was placed in a petri dish and left to dry for about 2 to 3 days. In bioplastics containing only PVA and flavonoids, the procedure is the same, obviously without the steps where starch and glycerol are added. In this way, PVA-based bioplastics were obtained with quercetin and bicalien up to the final concentrations of 0.5 and 1.0 millimolars and curcumin up to the final concentrations of 50 and 100 micromolars. Each bioplastic appeared macroscopically similar and homogeneous, with a different color depending on the total amount of polyphenol. At this point, the analysis of their functional and biological characteristics was performed. Unfortunately, already from the first tests carried out it emerged that bioplastics containing starch and glycerol were not suitable for our research purposes. So we abandoned their use and continued experiments with bioplastics containing only PVA and polyphenol. Morphofunctional analysis of the bioplastics was performed by means of FTR spectroscopy. As can be seen from the graph, the modification of the starting materials with the polyphenol doesn't induce the appearance of new bands due to the formation of covalent linkage with PVA, indicating a dispersion of utilized materials to functionalize the new sensors. It is possible to notice mainly the appearance of a broad and intense band at about 1600 to 1700 cm-1, characteristic of the functional group present in these natural compounds. In particular, they can be attributed to the greater vibration energy of the carbonyl group present in the identified flavonoids and to the water molecules bound. There are some changes also in the bands at about 1100 cm-1 influencing the sequence with crystalline and amorphous structures that are directly related to the modification of the PVA. Again, as can be seen, the scanning electron microscope images highlight little changes in the surface morphologies in the absence or in the presence of the tested compounds, with a general increase in roughness in this last case. This more heterogeneous structure can be attributed to the interaction between the polyphenol molecules and the macromolecules, which hindered the alignment of the polymer chains to form polymer-polymer intermolecular interactions, favoring the formation of a semi-crystalline material. Moreover, the analysis shows as the polyphenol are homogeneously dispersed in the polymeric matrix, according to the other test described in the above slides. Regarding the sensor-like properties, the newly synthesized materials were tested to verify their possible ability to detect metal ions present in solution, changing their color. They were placed in contact with solutions containing copper, zinc, iron, mercury, and nickel ions in the final concentration of 1.0 to 50.0 ppm. From the graph, we can see that quercetin is able to detect all the ions present in the solution, in particular copper and iron ions, while the effect is smaller in the presence of zinc, as it is evident from the change in color of the bioplastic highlighted by the colorimetric assay using the CIE lab scale, and particularly the delta. The same results can be seen for curcumin, which significantly changes its color in the presence of different ions added to the tested solutions, in particular zinc and iron. Bicalien changes color even in the presence of metals, although, as we can see from the graph, the colorimetric variation is not so markedly appreciated by the instrument. So from this colorimetric test, we can highlight the possible activity of bioplastic containing PVA and polyphenol as an environmental sensor for metal ions. In addition to their sensor capacity, spectrophotometric investigation can be used to study the possible bioplastic ability to purify aqueous solutions containing the metals. In fact, the interaction between metals and bioplastics monitored by spectrophotometric analysis using a probe capable of specifically detecting the presence of specific metal ions allowed a detailed study of the quantities of metal ions chelated by the different sensor used. This interaction causes a variation in the absorbance values at 620 nanometers that is evident already after 4 hours of incubation for all the flavonoids tested. From the experiments carried out in the presence of metal ions, it was evident that the synthesized bioplastics, in addition to acting as sensors for the metals tested so far, they also possess significant activity as adsorbents. Again, was also analyzed the ability to detect the presence of organic pollutants, crystal violet and phenol red, in solution. As it is possible to see from the graph, PVA and polyphenol-based sensors at different concentrations are able to decrease the amount of crystal violet present in solution, also resulting in a colorimetric variation of the sensor itself from yellow to violet. In particular, solutions containing crystal violet at a concentration of 0.8 milligrams per milliliter were placed in contact with PVA plus polyphenol sensor squares. After just 4 hours of incubation, a decrease in the absorbance values of the solution recorded at 594 nanometers was noted, in particular for the quercetin, which increased with the passage of time, up to 72 hours. This color shift of the sensors towards violet was quantitatively analyzed. In fact, in the graph is evident the transfer of the organic pollutants on the bioplastics and in particular the greater effectiveness as an absorbent if the polyphenol are linked to the PVA. Then we tested the sensor capacity to detect the presence of phenol red in solution. Spectrophotometric analysis of the supernatants taken from the test tubes containing the different bioplastic immersed in a 0.08 milligrams per milliliter solution of phenol red highlighted over the days a slight change in the absorbance values at 432 nanometers with all the substances tested, but at the same time an increase in the absorbance values at 560 nanometers was found in all the test tubes, which highlighted the color change from yellow to pink, much more marked in the presence of curcumin and completely absent in them. Test tube containing only phenol red in the absence of the sensor. The color shift of the sensors towards pink was confirmed and quantitatively analyzed by colorimeter study. So long as the new materials created, in addition to being sensors, are able to absorb pollutants, they could be used in bioremediation. Then, it was necessary to analyze the biocompatibility of the synthesized sensors, incubating a square of these last, with red blood cells at different time intervals, monitoring hemoglobin release, membrane integrity, methemoglobin presence, and SH groups titration. The results obtained after incubation of the polyphenol functionalized biopolymer with red blood cells show a reduction in the rate of hemolysis up to 6 days of incubation compared to the control in all the bioplastic with polyphenol and in particular in the presence of quercetin and bicalien. These data were confirmed by the morphological analysis of the cells, which shows a better membrane structure in RBCs incubated with bioplastics containing PVA functionalized with polyphenol compared to control. The observed beneficial effect by the polyphenol addition is confirmed by the evaluation of the oxidative state of the RBCs. The graph shows a reduction in the percentage of methemoglobin in presence of quercetin, bicalien, and curcumin compared to the control, even if the effect is more reduced in presence of curcumin, which is similar to PVA alone. In addition, higher levels of reduced thiol groups were observed in RBCs after 1 and 2 days of incubation with PVA with polyphenol compared to the control. Then, the results showed that the presence of polyphenol has a protective effect on the red blood cell. Analyzing the data, it also emerges that among the new created polymeric materials, although PVA alone is able to reduce the levels of hemolysis and methemoglobin compared to control, the ones with quercetin and bicalien incorporated to PVA showed the best results. Therefore, from the results obtained so far, we can affirm the complete biocompatibility of the PVA-based bioplastics functionalized with the polyphenol. In conclusion, the results showed that by structural and morphological point of view, SEM and FTR show change that support utilization of polyphenol to create this type of sensor, taking advantage on the basic skeleton of these molecules and of the substituents present in different positions. The synthesized sensors are able to analyze very low concentrations, ppm, of pollutants, both metal and organic ones. They are completely biocompatible. They can be easily utilized by a simple colorimeter and/or smart devices. I'm done. Thank you for your attention.